is pharmaphorum web editor Nicole Raleigh, and I am on site in Chicago for ASCO 2025, and with me are several EVERSANA experts. We're going to be discussing the Congress as a whole, and I'm going to let everyone introduce themselves, perhaps with a short description of what each of you do.
Hi, I'm Vanitha Sankaran. I am the lead medical strategist at the Oxygen affiliate of EVERSANA.
I'm Sarah Henry. I'm a medical strategist as well in the Oxygen affiliate.
Awesome. Tim Lewis. I am part of just the overall agency affiliate, agency business unit. I work on business level strategy.
And I'm Jordan Schell. I'm an associate partner within our management consulting practice.
Thanks, everyone. So, Tim, perhaps you can go first with the question liftoff, as it were, for this conversation. What's the most promising research you've seen presented at ASCO this year that could change practice within the next 2 to 3 years?
Yeah, great question. I'm gonna say the word, and then Sarah and Vanitha will talk a little bit about it. And I think I'll follow up with where we go after that.
Okay.
So not a big surprise. Biomarkers. Everyone's talking about it. Should be talking about it. I think you guys have seen some interesting papers over the presentations over the last couple of days. So why don't you start, and then I'll talk about maybe some ways we can expand from there.
Right, so there have been a number of papers presented in the last couple of days, like on biomarkers such as circulating tumor DNA, ctDNA in breast cancer, in bladder cancer, colorectal cancer. There was a very interesting talk on electrophysiologic biomarkers for diffuse gliomas, and also a set of talks that we both attended on DLL3 as a potential biomarker for neuroendocrine carcinoma. And what was really interesting that came out of these talks is that every different cancer has the potential for many different biomarkers, but how they present and what they mean to each individual patient can be really different. For example, in small cell lung cancer, DLL3 is highly expressed. So you don't really need to test for it. You can just use a DLL3 targeted therapy and know that most of your patients are going to respond. When you look at carcinoma outside of the lungs, though, DLL3 is expressed really well in some patients and less well in others. And in those patients that it's expressed really well, and unsurprisingly, a DLL3 targeted therapy works really well. You get great outcomes. In those patients that express DLL3 a little bit more poorly, you still get success though. You get some measure of disease control, meaning for those patients, you might take that DLL3 targeted therapy and add something else to it to get the efficacy that you want. So understanding, what biomarkers to test for, what patients might respond to what therapy is really going to help the oncologist make sense of all of the new therapies that are coming online. That is if we can get enough access and manage to pay for all the extensive testing that this kind of work is going to require.
And so some of the things that we've been looking at too in some of the talks along those lines, especially with therapeutic biomarkers, are now that there are combination therapies, triplet therapies, maybe even more, how do physicians sequence appropriately? A lot of these therapies have had really exciting efficacy and surprising safety. They're relatively safe and manageable. But now, how do you make the choice? How do you identify which patients are most likely to respond? And then what time? Do you use it early in the disease progression or later? And then one thing that we've been hearing a lot in talks as we're talking about all of these exciting biomarkers is for these therapies that are combination, what if patients become resistant to not one, but two of these really important targets for that particular cancer? Then what do you do? What's left for them? And so there's a lot of questions around how do we find the right patients using biomarkers and then sequence appropriately.
That just reminds me of a really exciting talk that I heard on a trispecific in the multiple myeloma space, where a lot of these multiple myeloma therapies are BCMA directed, but there's also the protein GPRC5D, which is generating a lot of excitement. And this trispecific targets BCMA, GPRC5D, and CD3, and it is showing better efficacy than a bispecific and much better safety. So it's very exciting, offers the potential to really go after all the multiple myeloma cells to get to MRD quickly and maybe have lasting remission. But what if a patient relapses? To Sarah's point, what do you give them next? Are they not responding to the BCMA, the GPRC5D? How do you suss that out? I mean, exciting work out here, but also prompts more work to be needed.
Yeah.
So on that note, when we think about biomarkers and we think about the next couple of years, one of the real questions you mentioned a minute ago, Sarah, is around sequencing. The biomarker pool will only get larger; it's not going to get smaller. And so we start to ask ourselves, what are true driver biomarkers, and how do we make decisions around that? How do we sequence? What do we think is the primary in any specific instance? One of the things that we could do probably easily is say, well, protein versus DNA. Probably we're going to look at DNA over protein if it's expressed, but that only solves a small part of the picture. So one of the things that we think that we'll see over the next several years, institutions like MD Anderson and Dana-Farber are jumping into what's the role of AI in helping us determine sequencing. We'll talk a bit about this perhaps in just a minute, but if I look over the next 2 to 3 years, one, it's going to be biomarkers. Two, the question around that's going to be sequencing. And then three, what's the role of AI in helping us determine that sequence? Because we'll never get enough studies to do that definitively. You'll never get enough head-to-heads. They'll always trail what's coming out as emerging biomarkers. We'll never get to that answer by looking at clinical trials by themselves. So what do we do in the interim, and what are the outcome measures? You mentioned MRD. We'll come back to this. What do we use as interim measures so we don't have to wait 3-4 years to get results when, again, biomarkers and new agents are coming out all the time?
Great, thanks, Tim.
You mentioned waiting 3-4 years. I've seen a lot more papers and posters around ctDNA and ctDNA clearance in the solid tumor space. So following the way of multiple myeloma, we're seeing in areas like breast and lung cancer, the use of ctDNA clearance to determine outcomes for patients. And what we've seen is improved overall survival. There was a poster around the EVOKE-1 trial for sacituzumab that actually showed increased anti-tumor activity with the clearance of ctDNA in that trial. So I think what we're going to start seeing is accelerated approvals using ctDNA as a surrogate biomarker in place of things like overall survival, and not waiting multiple years for these trials to read out.
Thank you for that, everyone. So we've discussed targeting, but I want to bring it back to AI and AI's role in targeting, because what is often lacking is certainty. So Tim, tell us what AI can offer in terms of certainty, just making it more certain.
Yeah, that's a great question. By the way, just as a quick aside, I think AI and certainty are words that are not often said together.
Yes, I realize that.
So
Yeah.
we're in a great spot to be able to say, how can AI provide certainty when patients' lives are on the line? I will take a quick moment to say one of the things that was just announced in the opening session is a partnership between ASCO and Google using Google Gemini with something that's called ASCO Assistant. And this is going to be an AI-driven approach to how do you get through ASCO guidelines, which can be complex. And in the past, AI has not been a great agent for this. This partnership, I think, will promise more. We've had really unfortunate circumstances like IBM Watson from the past that really were uncertain. I think this one provides a greater degree of certainty for clinicians as they're doing it. But let's talk about where we're seeing definitive AI wins today. I'll provide maybe two right now, and then we'll talk about certainty around primary outcomes. So there was an interesting presentation that was provided by MD Anderson, looking at ML and looking at it from the perspective of where 3 or more biomarkers are existent. Can AI provide a greater degree of certainty around outcomes using AI to make decisions versus tumor board? MD Anderson showed that for ML, they were able to show a hazard ratio of 0.28 on PFS using AI to drive what the treatment decisions were versus a tumor board. Really, really interesting data there. More to come from them, and more to come from other healthcare systems as well. But they're able to show this distinct benefit, trusting AI to make treatment decisions where it gets really complex with multiple biomarkers. So this biomarker thing continues to be a thing for us.
Yeah.
So that's MD Anderson. Dana-Farber showed some similar benefits, not directly to PFS, but in a different category. Those are really one that was an abstract, but those are really interesting places where AI is decluttering where things are getting more cluttered from multiple biomarkers. So I think there's a lot to be done there. I was in a talk a bit earlier on the neuro side, where AI is providing certainty around both how much to excise from a surgical perspective, and then what to treat as an adjuvant therapy coming out of that. There's so much going on from a neuro perspective around like you have to be precise, and so there's some really interesting things happening there. But I do want to come back if you don't mind. So AI, there's a lot to come. It will only get clearer, but those were two great examples from MD Anderson and Dana-Farber about places we can look. I do want to come back. You mentioned about ctDNA. The other part I think around certainty is how can we move outcomes earlier and provide greater certainty around what a therapy today can provide benefits, and what are initial markers that we could look at from that perspective. So you mentioned ctDNA. I think there's a lot happening there. Maybe we can talk about that again for a minute. But also the idea of MRD. You mentioned MRD a little bit earlier. So we now know the FDA has accepted MRD as an outcome measure, a primary outcome measure. Awesome. And I think that the idea there is, can we truncate 2, 3, 4 years of data that it would take to typically get down to something that you could get in months versus years. I think ctDNA is another example of that. With all of the therapies that are coming out, with all the biomarkers that are coming out, if we can truncate the amount of time it takes to reach a decision that this is working, and it's an indicator of it working long term, those two are great measures for us. And I think that's a place where trial designs need to change for both of those. And I think that there'll be a lot of confidence driven as well. So not AI specifically, although AI may come into play with that, but really how do we truncate to get to certainty earlier, whether that's AI, whether it's these new emerging markers of outcome.
Great.
I think that's really interesting. I think that AI certainly has the potential to help us get drugs to market more quickly. One of the areas that I think we really need to think about is how the FDA is changing what kinds of data that they'd like to see.
Yep.
In the lymphoma space, they recently made the decision to reject a therapy moving up earlier to early line setting because not enough patients were enrolled in clinical trials in the US. And so how can we do exactly what you're saying, use AI to find those markers that'll bring those therapies forward, but also convince the FDA that we are picking the right patients in our clinical trial sites to show that we are doing robust clinical studies. And that's something that we have to watch out for because, every administration, every era has, new ideology from the FDA. And how do we bring that into it as well?
So this caution and these changing times, in that journey, you hear a lot of hype and you also hear a lot of lack of hype. So I was wondering, for each of you at ASCO 2025, what in your opinion has perhaps been slightly overhyped, if you're willing to share that, and also what in your opinion has been underappreciated?
One thing I've been hearing a lot too, and we've been talking about, I don't know if it's underhyped, but the patient journey is so important with a lot of these really difficult treatments, bispecifics, CAR T. There's a lot of management that needs to happen, and patients need to be followed and fully understanding that journey. Travel to the clinic, travel to a hospital, needing a caregiver, things like that. We're definitely talking about it, but the total understanding of what a patient goes through for some of these therapies, we're still figuring out and still trying to figure out the models that'll work in a really smart way to make sure patients don't have side effects that cause them to go to the ER and things like that.
I think it's a great, I'm going to build off that just for a second. I think it's a great example of as the supply chain has gotten figured out from multiple companies now on the CAR T side, we were at a place where there was not enough supply for demand, and so you didn't have to worry about some of those things. Now that there isn't that sort of supply, and there are so many more CAR T therapies coming out, I think you're exactly right that the idea of we have to think differently about how we manage patients when there's many more patients that could benefit from it, especially to make sure that patients do get it. So I think that underappreciation of patient journey makes a lot of sense to me.
So I've been thinking about this question, and we spent a lot of time talking about AI. And I'm going to take the position that I actually think that it's also underhyped from our friends in the pharmaceutical industry, right, who are manufacturing the products. We've talked about inclusion of things like ctDNA and MRD in clinical trial design. but we also can see in some poster presentations that have come out in this ASCO where the use of things like AI in determining prognostic factors of overall survival or progression-free survival has actually had a very big impact in predicting how patients are going to respond on certain therapies. There was a Cleveland Clinic poster that I saw in resectable NSCLC where the use of radiomics actually was able to predict outcomes better than PD-L1 expression alone. And I think that we're at the precipice now where we can start including some of these new existing technologies into trial to further accelerate approvals or our understanding of biology of some of these new products.
So final question. For everyone here, if you could solve one major problem within oncology over the next decade, what would it be?
For me, it is access. Therapies are not getting less expensive, and we're probably justifying the cost. So the challenge for me with regard to access, treatments are gonna reach smaller and smaller patient populations. Generally, when that happens, the cost gets bigger and bigger and bigger. And so for me, there is a real access a challenge to solve around how do I get more personalized, and I also get less financially toxic for patients, and I'm able to reach all the patients that should deserve that therapy. I think access is a tremendous one over the next decade.
Absolutely.
I think going along with the idea of access is looking at those patient populations that are typically really underserved already by the health system. You know, we were talking about, again, in multiple myeloma, how we have CAR Ts coming online for that, new CAR Ts coming online. And, you know, particularly in the multiple myeloma space, that disease predominantly affects the Black community. But there are very few, if any, Black CAR T prescribers, CAR T prescribing and treating doctors.
I'll stick with the theme of access as well. But what we're seeing is new advanced therapies like bispecifics, trispecifics, different T cell engagers, CAR Ts, moving not just into the heme space, but also into the solid tumor space. Most patients are treated at a community hospital setting in the United States, vast majority. But most of these treatments are only available in academic settings where a lot of these patients, like the Black community, struggles to get access to. And so how do we activate these smaller community centers or larger cancer networks to start adopting these therapies sooner so we can actually bring them to more patients that need them?
I was going to touch, I know we talked about it already, sequencing, but I still think I heard a great intro to one of my sessions that talked about, don't quote me, but that last year there were 14 new approvals, FDA approvals in oncology, and already this year there have been 4. So the rapid pace of innovation and development is wonderful in oncology, but has a consistent need for everything that we've been talking about in order to help guide physicians, both in the academic community, but in the academic setting and in community, to understand when to use treatments. Now that there aren't really many disease states left that don't have therapies, which is wonderful. Most have several. Now, how do you choose and make the right selection in a really smart way, including access, including patient journey? It's a really difficult process, but really exciting too.
Exciting indeed.